Good evening to all the CUET aspirants. So here we are in our third session of Phycology, where in previous sessions we have dealt with the complete character life followed by chlorophycy in detail. Also to the type of cycles detail where we size, box and this unit are good. Today we are going to learn in detail about two more classes of algae, and rhodophyceae. First, looking at salient features of phycophyceae, the brown algae. This phycophycean member is also known as brown algae. This is due to what pigment? Fucoxanthin pigment. This xanthan pigment. This two genera and 15,000 species and globally with 31 genera and 141 species reported from the Indian coasts. Mostly the brown algae are marine. They are found to be in temperate and cold water regions in Lithosphere. For fresh and Plurocladia, Spacilaria. These are different species which are in freshwater genera. Okay, and the cell is composed of, like cell wall has two layers in the brown algal members. One is cellulosic layer, inner cellulosic layer, and outer layer made up of inner is cellulose, layer is pectin. Also has composition of alginic acid and fucoidin. Alginic acid and fucoidin. In intercellular spaces, calcification is padina. Okay, calcification is observed in padina. Then chloroplasts are discoidal and ellipsoidal with 3 thylakoid bands. Okay, 3 thylakoids per band. How many chloroplasts are there? Chloroplast shape is discoidal and ellipsoidal, and in each chloroplast, how many 3 thylakoids present as band. Okay, this is surrounded by chloroplast top. CER membranes. What is CER? Chloroplast endoplasmic reticulum. Okay, this is characteristic feature of brown algal members, where the outer membrane is continuous. Outer membrane of this is continuous with the nuclear membrane. Fine, right? Outer membrane of chloroplast endoplasmic reticulum membranes, they are continuous with nuclear membrane. And pyrenoids. Pyrenoids are the food storage organelles. These are present inside the chloroplast. Okay, pyrenoids, they are single naked and stalked structures. They produce from the chloroplast. Fine, right? These are the salient features of phycophycean members. Clear till here? Any doubts anywhere? Yeah. Then photosynthetic pigments. What are the major photosynthetic pigments that you can observe in brown algae? Photosynthetic major photosynthetic pigment is chlorophyll A and chlorophyll C. Chlorophyll A and C are the photosynthetic pigment, but the coloration from green to various shades of brown color is given by fucoxanthin. Fucoxanthin will give the colors from olive green to various shades of brown color. Olive green to various shades of brown color. Here cells, reserve food material is laminarin and mantle. Laminarin and mantle are the reserve food materials. Cells here are uninuclear, prominent nucleoli at the center. Okay, cells inside them, they have prominent nucleus, which is uninucleate condition, and it has prominent nucleoli. And here if you see motile cells, these are gametes and zoospores. They will be having one long anterior prehensile flagella, okay, in pantonymatic flagella, posterior whiplash flagella, long anterior prehensile flagella and short flagella. Hair mastigonines over it, and there is an eye spot which is present. This will aid in phototaxis. What is phototaxis? Movement with respect to light. Okay, the movement in accordance with light is known as phototaxis. Now what is about fucosane vesicles and these are discovered by Schmidt 1888. This is discovered in the year 1888, which are present, and they contain protannins, and they oxidize to form phycofine. Okay, they oxidize to form phycofine. Fucosane vesicles or physodes, they are discovered by Schmidt, and they contain fluorines and phycofine. Thallus type here. Thallus type, they range from heterotrichous filamentous to true parenchymatous forms. Okay, thallus form range from heterotrichous filamentous forms to true parenchymatous forms. And there is no unicellular or simple filamentous thalli. There is no unicellular or simple filamentous thalli. Vegetative reproduction. The common mode of vegetative reproduction is always fragmentation. Common mode of vegetative reproduction is always fragmentation. Asexual reproduction is by zoospores, by unilocular sporangia, which undergoes meiosis, and this produces haploid zoospores. Next, plurilocular sporangia and tetra in some genera. We come across two types of sporangia. One and unilocular sporangia. Plurilocular sporangia are present at the terminal ends. We will discuss about it in detail in the coming slides. Next, sexual reproduction, mostly isogamous and anisogamous types of sexual reproductions. Okay, sexual reproduction is mostly isogamous and anisogamous type. Alternation we can heteromorphic type of alternation of generation observed. We can come across diploid life cycles. So this is a complete, this slide, entire slide is important. Every point is important for your exam. Now we will move on to a type study. First is about ectocarpus. Where is the ectocarpus actually present? Occurrence. These are widely distributed in the marine algae. Widely they are distributed. Marine algae is found both in the tropical and temperate oceans. Marine algae is found both in the tropical and temperate oceans. This grows as epiphyte. This will grow as epiphyte on the other plants. And epilithic tufts means they will grow on the rocks. Okay, epiphyte. The ectocarpus is growing on the plant surfaces, and epilithic, the ectocarpus growing over the rock surfaces. They are found in the littoral and sublittoral zones. Okay, they are found both in the littoral and sublittoral zones. Thallus structure. What is the thallus structure over here? Thallus is both branched. Branched we can see of two types of branching. One is filamentous branching, where single filament with single row is present, and next is heterotrichous branching. Okay, multiseriate also we will call it as, or heterotrichous branching also we can call it as, multiseriate or heterotrichous branching. Heterotrichous branching. This will differentiate into prostrate filaments and erect filaments. Erect filaments are more, prostrate filaments and erect filaments are more branched. Branch is below the septa and end. Okay, branches arise below the septa and usually they end in head. Rhizoids are from lower branches and they attach to thallus. Rhizoids are from lower branches and they attach to thallus below the surface. This will contain ribbon-like or plate-like plate-like chloroplasts. Okay, Ribbon-like and plate-like chloroplasts are, they are without pyrenoids, with or without pyrenoids, and end branches are long with tapering ends. They have hyaline and they do not have chloroplasts in them. Cells are uninucleate. Cells are uninucleate with mature cells having large vacuoles. They will be having large vacuoles formed by fusion of small vesicles. Large vacuoles are formed by the fusion of small vesicles. This is the complete thallus description of ectocarpus. It is branched, it can be uniseriate or multiseriate, and branches, one is prostrate filamentous branching, next will be erect branching, two types of branches. Branches will arise below the septa and end like hair-like structures. They end with hair-like structures. Rhizoids, which are formed at the base of substrata, and the cells will contain ribbon-like structures. Ribbon-like structures are plate chloroplast. May present or absent inside the chloroplast. End branches will have tapering ends. Cells will be uninucleate mostly, but large vacuoles, they are formed by cell fusion. This is the basic description of ectocarpus thallus. This occurs while zoospores, where are they formed? They are formed in sporangia. Sporangia we have to see. Okay. Sporangia. One is plurilocular, second is unilocular sporangia. Plurilocular sporangia is elongated. Okay. Plurilocular sporangia, here you can see these are elongated cells. Unilocular sporangia will be globose shaped or it can be ellipsoid shape. Here we see first they develop on the terminal cells of the lateral branches. Plurilocular sporangia, they develop on the terminal cells of the lateral branches. Mitosis will divide. Okay, mitosis divides the protoplast into two vertical rows of cuboidal locules. Each locule will have, they form a pear-shaped diploid zoospore with two unequal laterally inserted flagella. Zoospores are then released through a pore. They swim briefly and they develop into sporophytic thallus. They swim briefly and they develop into sporophytic thallus. This is about plurilocular sporangia. Okay, wherein they develop on terminal cells of lateral branches. Mitosis will divide the protoplast into vertical rows. First cuboidal locules are formed. Each locule will have diploid zoospores which are of pear shaped and they have two unequal flagella situated at the lateral ends. Unequal flagella are present. Now zoospores are then released through pore which they develop into what? Sporophytic thallus. This is about plurilocular sporangia. Now look into unilocular sporangia. Unilocular sporangia, here spores will undergo meiosis followed by mitosis. Spores undergo meiosis which is followed by mitosis. This is to produce 32 to 64 haploid nuclei are formed. Here you can see diploid zoospores in the plurilocular sporangia. Haploid nuclei are formed. 32 to 64 haploid nuclei are formed immediately after meiosis. Each nucleus and protoplast forms haploid biflagellated pear-shaped zoospore. Remember, the shapes of the zoospores must be clear both for plurilocular and unilocular. Haploid biflagellated pear-shaped zoospore. These are liberated and they develop into.
Okay, so next point is
they'll get developed into gametophytes. This is about the asexual reproduction.
So Q is
unilocular sporangia. Then
negative. Okay. So
sexual reproduction. Types of sexual reproduction you see. Isogamous and anisogamous type of sexual.
Q by something.
Some species, for example, here you see Ectocarpus siliculosus. Ectocarpus siliculosus are heterothallic. The heterophytic thalli will produce plurilocular sporangia or gametangia, which is similar to that of sporophytes. Each locule, inside each locule a gamete is formed. This gamete is morphologically similar to zoospores. In physiological and physiological
specific key and
morphologically it is same, but physiologically the gametes will be different.
so
Gametes, they act as passive female gametes, others are
same
motile male gametes. Two types of gametes are observed. Morphologically they are same, but physiologically it will be different. In Ectocarpus sedans, in Ectocarpus secundus, female gametangia are large. Female gametangia are which are producing large.
okay. No.
Female gametangia is producing large female gametes. Female gametes will secrete ectocarpin. Ectocarpin is a sex hormone. This will attract the male gametes. Multiple male gametes will surround around one female. The hormone is very important. What is female gamete? Ectocarpin. Fine, right? Around which many male gametes will be surrounded. One will fuse via apical ends to form what? A zygote. This zygote will develop into what? A new sporophytic thallus. Unfused gametes will germinate into what? Gametophyte. Gametes will sporophytic thallus. So this is about sexual reproduction. From asexual reproduction, we have learnt about two types of sporangia. One is unilocular sporangia, second is plurilocular sporangia. Sporangias, their shapes, how zoospore is, in which type of sporangia process is happening, in which type of these things should be addressed carefully.
Because of the surrounding, it becomes cool.
So, second is sexual reproduction. Sexual reproduction is both by isogamy and anisogamy. Okay, sexual reproduction
In the surrounding,
isogamy and anisogamy. If it is isogamous, morphological isogamy we have observed and physiologically anisogamy. Example you see ectocarpus. And you observe larger than male, where it is producing a hormone called as ectocarpin. So ectocarpin is attracting the male gametes, and after fusion, post-gametangial fusion, zygote is formed, which develops into a sporophytic thallus. The unfused male and female gametes will germinate into a new gametophytic structures. Here it is clear, right? Yeah. This life cycle. Life cycle shows formation of generation, where you can see equal haplontic and diplontic life cycles. Okay, this shows diplontic life cycle, ectocarpus. Haplo.
The mixture, temperature of mixture. You can take a note of this.
Life cycle. A diplontic life cycle. Here you observe zoospores which are formed, which are for the main sporophyte diploid. Sporophyte which is diploid. Okay. A portion of the sporophyte, if we have to observe, first we will look into the asexual reproduction. Plurilocular sporangia is also diploid. Plurilocular, unilocular. Later might happen diplontic plurilocular sporangia after mitosis. Okay, unequal flagella are present inside each zoospore, which is again diploid. Biflagellated zoospores are formed. They will germinate into a diploid sporophyte. So it is completely diploid. Unilocular sporangium in sexual phase. In asexual phase, plurilocular sporangia. In sexual phase, unilocular sporangia which is diploid. Inside unilocular sporangia, after meiotic division, 32 to 36 gametes are formed which are haploid. You can see zoomeospore which is haploid. Biflagellated zoospores formed. This germinates into haploid gametophyte. Okay, haploid gametophyte later matures to plurilocular gametangium. Later, after gametic fusion, which is N plus N condition, after gametic fusion, this forms diploid zygote. Okay, after gametic fusion, it forms diploid zygote, which is sporophyte. Later, zygote matures to diploid sporophyte. Clear till here? Any doubt anywhere? Yeah. And then life cycle shows isomorphic alternation of generations. Gametophytes are dioecious, bearing only prelocular gametangia.
What happens here? The heat has been transferred from hot to cold.
So
the heat
asexual cycle, this produces diploid zoospores. Okay, plurilocular produces diploid zoospores. Here, zoospores form unilocular sporangia. This develops into gametes. Zoospores form plurilocular sporangia. This continues sporophytic generation. Clear with this? Next. Next is type study that we are going to see is Dictyota. Where is this Dictyota observed? Dictyota, it is found over rock pools, which is epilithic of littoral and sublittoral regions of marine water. Mostly part of Dictyota is marine water only. Widely these are distributed in tropical, subtropical and temperate regions. Thallus, if you see for Dictyota, it is flattened, bushy, erect. The peculiar feature or the characteristic feature of Dictyota, if you observe, is dichotomous branching. Dichotomous branching is the characteristic feature of the Dictyota. Okay, can you see here dichotomous branching? Okay, dichotomous branching is the characteristic feature of the Dictyota. But this dichotomous branching, this can be pinnate in same species. When attached to the substratum, this will be attached by tuft of rhizoids, that is group of rhizoids. This will be attached by tuft of rhizoids or group of rhizoids. Okay. Growth occurs by apical lenticular cell, which is located at the tip of each frond. This is located at the tip of each frond. Apical cell cuts the segment basically which broaden from the apex to base. Each segment will divide into two curved walls. First, a larger inner medullary cell and two layers of small cortical cells. Medullary cells and smaller cortical cells. In mature cells, you can see medullary layer, large cells with few or no chloroplasts. If you see, they are small cells which are densely packed with chloroplasts. Small cells with densely packed chloroplasts. True hairs will arise as tufts and they are scattered on the surface. Branching occurs by equal vertical longitudinal division. Okay, they are by equal vertical longitudinal divisions. This is how the branching occurs. Fine. So mature thallus will be having medullary layer and cortical layers. True hairs will arise as tufts on the cortical layers. This is complete thallus description of Dictyota. Now about sexual and asexual cycles. Sexual and asexual stages. Vegetative reproduction, this occurs by detachment of frond portions. Okay, vegetative reproduction occurs portion.
So we'll get
The piece will later regenerate into what? New thalli. Sexual reproduction, here you observe reproductive structures, they appear as sori or patches. Reproductive structures are sori or patches on the lower parts or on the margins of the fronds. They appear as parts or the margins of the fronds. And tidal rhythms will influence the sexual reproduction. In Dictyota dichotoma, gamete develops into neat tidal periods in summer. Formation of sporangia is not linked with tidal periodicity. Here male gametophytes, they bear plurilocular sporangia.
right now, I guess.
where sori are present on the surface of fronds. Vegetative cells near sori, they divide to form a protective covering is formed. Each chamber of antheridium will produce one male. Each this not
done.
oosphere.
So
Oosphere.
we are entering
This matrix.
solid to liquid.
Eventually, oosphere is free.
There is no state change.
After
Right now, state change is nothing but going from
the excel.
not like that.
Later, after fusion, post
To gas like
what is formed? What is formed which dip.
from one state
This attaches to the substratum and germinates into a new
solid to liquid.
new sporophyte which is
So what we
diploid. Fine, right? So here male gametophyte and female gametophyte.
boiling right now.
The gametophyte will produce and by
If I start melting the ice, it becomes.
they are produced inside the sori.
If I start again heating that, it becomes
Right? It has a protective covering to this chamber.
right.
Here they are
State. Okay.
each
Try to adjust.
will produce one tile female.
So melting, the change of state from solid to liquid. So melting point is nothing but
Condition.
at which solid and liquid state of the substance is in thermal
To a new sporophyte.
in the sense like
Clear?
solid
Then
like amount
you
of
can cycle
ice, complete water, but some there is no thermal equilibrium. Okay, that's it. We are talking.
heteromorphic alternation
State from
is observed where macroscopic sporophyte is seen. This sporophyte produce haploid zoospores.
nothing
Meiosis.
the
Zoospores they form
liquid and the vapor
post fertilization that
thermal equilibrium. Boiling point. One more is melting point. Change of
so yes. With this we felt up till dictyota.
when we
Next class
talk about dry
we will be looking into
if I take
the rhodophyte,
it becomes
its general characteristics and typical features.
applications.
Rhodophycy general characteristics, example of rhodophycy
The phenomena of freezing.
birds where we will be looking into
Freezing. Okay, refreezing. Okay, so take a slab of ice and
with a-
wire and fix
we'll
to
be finishing
Okay.
mycology. Coming up we will be looking into mycology.
applications. Keep freezing.